Hi, my name is Olivia Osborne. I'm a postdoctoral research scholar at UCLA, working in Theme 2. I did my PhD at the University of Exeter in the southwest of England, specialising in ecotoxicology. And prior to that, I read biological sciences also at the University of Exeter. My project is actually the hazard assessment of nanomaterials, specifically in the environment and river systems. So I look at silver nanoparticles currently in my project, and that's mainly because silver is a hot topic of research interest, because a lot of consumer products have silver within them, and we believe that they might be able to reach river systems as a result. So I look at specifically at the zebrafish. So I use the zebrafish as a model organism from embryo all through to adult to be able to assess the hazard assessment of silver nanoparticles. The zebrafish is a highly versatile model organism. So for a start, adults can lay thousands of embryos daily, which give you a lot of possibilities for screening. The other reason is that they have a sequenced genome, which then means that you can create transgenic lines as well. But the main reason that people use them is because their embryos are very fast developing, and their chorion, i.e. the shell on the outer membrane, is transparent, which means that visible observations can be made very easily. So in line with the theme of the lab, that changing the shape or the size of the nanomaterial changes the behavioural outcome, I've recently discovered that changing the size of the nanomaterial drastically changes the toxicity outcome in the zebrafish. So our work is important from an environmental perspective and human health. Specifically though, my project, the environment benefits. As I mentioned earlier, silver currently is a hot topic of interest. So for us, being able to assess the hazard potential of this for the environment means then we can manage it in a sustainable way by product by safer design. So the reason for me to pursue science, and specifically my area of topic, which is the environment aspect, is because I love nature and I want to protect it. And one of my first experiences was actually at university, where in my first year of university in summer I went to the jungles of Honduras and I did terrestrial conservation work. And then the year after that I decided to pursue marine conservation work in the Bahamas. And although these were very exciting adventures, I actually didn't find a passion until I found myself in the lab working in a research project, which came from me having a position as a research assistant in the aquarium. And thanks to my wonderful supervisors, I was able to combine two of my favourite subjects at university, which were developmental biology and ecotoxicology. And as a result, I am still working on those topics today. This took place in my back garden in England, in a pond that my father set up. And I will call it the frog experiment. So I was fascinated to see from when the frog lays its eggs, which is essentially a ball of black dots, that then form into tadpoles, these little black strings, that then form legs and have gills, and they swim around. And then a week later, the gills drop off and the frog crawls its way out of the pond and becomes a fully functioning frog. And my father told me that then it doesn't have gills, but it actually breathes through its skin. This completely blew my mind, and actually then completely sparked an interest in developmental biology.